Okay, this is the Professor, CBMD PhD, and today's Wednesday, October 11th, 2023. It is 3:48 and 9 seconds, a.m. Eastern Standard Time, and today's research, pancreatic cancer. So let's jump to it here. Newfound mechanism suggests drug combination can start pancreatic cancer. Date October 9, 2023. Source, NYU, Langwood Health. Normally supplied by the bloodstream, oxygen, blood sugar, and other resources become scarce as the increasing density of fast-growing pancreatic tumors cuts off their own blood supply in the environment. The ability to switch fuels contributes to the deadliness of pancreatic cancer. The new study involves a drug designed to prevent pancreatic ductal adenocarcinoma cells from one such switch. Pancreatic ductal adenocarcinoma cells use the enzyme glutaminase to convert the amino acid glutamate into glutamine, a form that can be burned for fuel to sustain rapid tumor growth. Drugs designed to block glutaminase, however, have been shown to cause cancer cells to switch to still other scavenging pathways. And for this reason, the field next looked at experimental treatments like DRP-104, designed by a new prodrug formula compound 6-diazo-5-oxo-L-noroleucine that is preferentially activated within tumors to overcome toxicity issues seen with diazo-oxynoroleucine. As diazo-oxynoroleucine was designed to starve cancer cells by mimicking glutamine, which unlike glutaminase blockers, broadly inhibits all metabolic pathways that use glutamine. Currently in clinical trials against non-small cell lung cancer, DRP-104 cannot be burned as fuel, but clings to the same enzymes as glutamine. In the current study, DRP-104 treatment alone decreased pancreatic ductal adenocarcinoma growth in mouse models of pancreatic cancer. Importantly, the current team also found that pancreatic ductal adenocarcinoma cells pushed into metabolic crisis by DRP-104 increased signaling through a protein called extracellular signal regulated kinase, or ERK, to make up for their loss of glutamine metabolism. When the team combined DRP-104 with an existing drug that blocks the ERK signaling pathway tramtinib, it further improved survival in pancreatic cancer mouse models compared to DRP-104 treatment alone. As despite a decade of advances in understanding how cancer cells switch fuel sources, we have not yet effectively translated this into clinically relevant therapies. As broad antagonism of metabolic pathways with glutamine analogs may provide another mode of attack against these highly resistant tumor cells, the fact that such drugs are already being tested in the clinical makes us hopeful that we may finally see patient outcomes improve if this approach proves to be effective in clinical trials. Moving forward, the research team will seek to understand how glutamine antagonism impacts other adaptive nutrient scavenging mechanisms in pancreatic cancers and whether these can be targeted as well. The success of such approaches will depend on careful balancing of improved therapeutic efficacy and toxicity from the potential effects on normal tissues. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.